This next group of drugs are loosely defined as metabolic altering drugs or drugs that can inhibit gene expression. Okay, and so uh, from a mechanistic standpoint, there's several. A lot of these are kind of bordering on chemotherapy, at least like kind of classic chemotherapy. So of course, glucocorticoids or your steroids severely alter gene expression. They change the expression of upwards of 30 to maybe even 40 different genes. Some of these other drugs are also kind of classified as cytotoxic agents, and of course the standardized um, chemotherapeutic drugs listed here are cytotoxic. Anti-metabolites are and or alkylating agents, including azathioprine, mycophenolate, um, are your classic kind of inhibitors of metabolism in specific cells that have high turnover, such as cancer cells, but also immune cells as well. Methotrexate, leflunomide, cyclophosphamide, these are all kind of lumped in this category. So we're going to examine just a handful of drugs that have anti-metabolite functions, and they all have a very similar kind of mechanism. They basically get incorporated into either DNA or RNA or both, and they really kind of shut down homeostasis. Okay. Um, Enteric-coated mycophenolate has kind of replaced azathioprine because it's a much better safety profile. But basically, it gets kind of incorporated into the into the structure right here, which is biologically active. And officially, it's a selective, non-competitive, reversible inhibitor of the enzyme inosine monophosphate dehydrogenase. What does that mean? Basically, you shut down de novo synthesis of GMP. And GMP is really important for lymphocyte proliferation, um, production of antibodies, the process itself of cell adhesion and cell migration. So we're talking about immune cells, specifically lymphocytes. The pharmacokinetics is listed there. You can read that by yourself. Therapeutically, we can see ECMPS for prophylaxis of transplant. We'll combine it with steroids and calcineurin inhibitors, not with azathioprine because the mechanism is so similar. Toxicities are listed there. Lots of drug interactions, and don't worry about the dosing. Azathioprine is kind of the older one that gets converted into 6-mercaptopurine, and this really inhibits de novo synthesis. De novo, by the way, is Latin for from new or from scratch. So it inhibits from new synthesis of purine. Okay, and what does all this mean? Really, you're arresting cell proliferation, and it has a marked increase, a marked effect on lymphocytes because there's a lot of proliferation and turnover in lymphocytes. We'll see this drug used in transplant rejection, some other kind of inflammatory type diseases, rheumatoid arthritis, I think celiac or Crohn's disease, and probably a few others. Okay. Methotrexate is one of the folate analogs. It inhibits dihydrofolate reductase, causes apoptosis of activated or effector CD4 and CD8 T cells. Leflunomide shuts down pyrimidine synthesis, has a dramatic effect on B cells. That's why you can see it approved for rheumatoid arthritis, but it's also kind of being looked at in terms of lupus as well as myasthenia gravis, which have a antibody promoting effect in them. So there's an antibody promoting pathogenic application there. And so we want to shut down those antibodies. Cyclophosphamide is kind of your more potent chemotherapeutic drug that is immunosuppressive by nature. It alkylates the DNA. It kind of gets incorporated into the DNA and reduces the structural stability of the molecule. This will again have a profound effect on B cells, but ironically, it enhances the T cell response. Really, it's kind of getting at the marginal zones within the secondary lymphoid tissue. We don't have to get into that mechanism. One, I can't remember it, and two, it's not that important. But it does have a primary B cell inhibitory effect, moreover than a T cell effect. And then adverse effects are listed there. There's another group of drugs that can actually reduce immune cell movement. For simplicity's sake, I really just kind of focused on one, fingolimod. This is a sphingosine 1 phosphate receptor partial agonist. This one has a lot of utility in MS. And what it does is it basically sequesters immune cells, lymphocytes in particular, to the secondary lymphatic tissue, and it blocks their movement out of it. So you can see it as part of adjuvant therapy in graft versus host disease. It's clinically approved for MS. In terms of toxicities, there's really few, aside from having dramatic reduction in leukocytes and lymphocytes. You can see bradycardia, which manifests itself within 48 hours. That usually returns to normal. There's some negative chronotropy. Again, that usually returns to normal. So the key component here is that when you stop giving the drug, the immunity returns to normal. And so the concept here is that if it's the T cell population of cells that are going into a specific organ or a specific compartment, and they're kind of exacerbating immunity, you can kind of sequester them and shut them down by basically keeping them trapped in here. So they can't produce damage, the T cells can't produce damage in terms of the inflamed areas if they're basically locked away in this secondary lymphoid tissue, which could also be a Peyer's patch as well. All right, so that's kind of the last group of drugs that we're going to talk about. Let's look at some of these review slides to kind of put it all together.